having looked at how triploids can be created and how meiosis occurs in triploids, let's consider how tetraploids can be created. Tetraploids can occur naturally due to a failure of segregation of the chromosomes, and this can happen in nature, but it can also be, tetraploidy can be induced by treating with a chemical called colchicine. And what colchicine does is it prevents the polymerization of tubulin. So recall that during anaphase in mitosis, the chromosomes segregate, are pulled to the opposite poles by centrosomes, which connect to the centromeres of the segregating chromosomes by spindles. And these spindles are made of tubulin, and the formation of these spindle fibers requires polymerization of the tubulin. Therefore, if you treat the cell or the tissues during mitosis, so this is not about gamete formation in meiosis, but in somatic tissues during normal mitosis, during tissue growth, if you treat the cells with colchicine, it prevents the formation of the spindle fibers, and that results in an inability of the chromosomes to segregate. So for example, if we are looking at the cell in telophase or during cytokinesis, what will end up happening is all 4 chromosomes might segregate to one side, to one of the daughter cells, and thereby double the chromosome number, so that over here you had 2n chromosomes, but you end up with 4n chromosomes. And if these tissues, and this is most common in plants, give rise to germ tissue. So let's say this develops into a flower, then those gametes will be diploid. And when mated or crossed to other diploid gametes, will give rise to tetraploid organisms. One thing to note is how one would write genotypes in tetraploids. So let's say this individual was a diploid and a het, so that you have big A, little a, and after mitosis, with colchicine treatment, we will get a cell that has 2 big A alleles and 2 little a alleles, instead of receiving one of each allele. And we would write the genotype of this individual as A, little a. Next, let's consider how meiosis occurs in a tetraploid. Now, we will have at prophase 1 4 pairs of sister chromatids or 4 dyads, and in order to make it easy to track these different dyads, I will draw the second pair in a different color, but they're all the same chromosomes. So these are 4 non-sister chromatids, and they're all paired up in pairs of sister chromatids. Now, when we think about how these might pair during prophase, there are 3 possibilities. The first one is that we could just have 2 bivalents. And these bivalents will segregate to opposite poles, so that at the end of meiosis 1, we will have one pair in one daughter cell and the other pair in the other daughter cell, resulting in, after completion of meiosis 2, in diploid gametes. So this situation was one where we had 2 bivalents. Next, another possibility is that we have a quadrivalent. So all 4 chromosomes pair up. And so we have sort of 4 of them paired up together. But when we consider segregation, each pair will segregate to opposite ends of the opposite poles, and we will once again end up with a situation of two chromosomes or 2 dyads on either side, resulting in diploid gametes. And so this is 1 quadrivalent. A final third possibility is that 3 of the chromosomes pair up as a trivalent, and 1 chromosome remains separate as a univalent. And so we will label this as 1 trivalent plus 1 univalent. Now, when we are considering how they're going to segregate, this pair of the paired chromatids in the trivalent will go to opposite poles, whereas the third unpaired in the trivalent will go randomly to either side, as will the univalent, which will go randomly to either side. This could result in 2 different situations. One is, of course, that we could have 2 chromosomes on either side, and that would lead to diploid gametes, euploid gametes. However, since the 2 reds are segregating randomly, we could end up with a situation where you get 3 chromatids on one side and only 1 chromatid on the other side, and this would result in aneuploid gametes. So this would result in a 3N or triploid gamete, and this one will result in an N or haploid gamete. Now, the main difference between meiosis in triploids and meiosis in tetraploids is that in triploids, most of the gametes are like this. Most of the gametes are aneuploid, either triploid or haploid. However, in meiosis in tetraploids, a good proportion of gametes are actually diploid, and therefore, and diploid not just for one chromosome, but diploid for all the chromosomes, and therefore usually result in viable offspring and can be propagated as tetraploids. While autopolyploids can be created by chemical treatment or can occur naturally in nature by doubling the set of chromosomes from the same species, an allopolyploid is a hybrid between two different species, and that has more than 2 copies of each of the input genomes, and can therefore propagate as a polyploid. An example is an attempt by Georgy Karpachenko to create a hybrid between cabbage or brassica and radish plants, raffinus plants, in order to obtain a hybrid with leaves of the cabbage and roots of the radish. Now, both brassica and raffinus have a haploid number of 9, and they're both diploid species. However, the chromosomes between brassica and raffinus are called homologous, which means they are similar but not exactly the same. And when you cross them, the gametes of a brassica have N1 chromosomes, and the gametes of raffinus also have 9 N2 chromosomes, so that when these two are crossed, the progeny has N1 plus N2 is equal to 18 chromosomes. However, this new plant, although it's viable, is unable to produce progeny, is infertile. And the reason for that is that the chromosomes are homologous. And what that means is that, let's say I have a pair of sister chromatids from brassica, and I have another pair of sister chromatids from raffinus. These are not going to synapse or pair up during prophase of meiosis 1, since they are not identical, or they're dissimilar enough that synapse will not be possible. And that implies that in anaphase, these chromosomes could segregate to either side independently of each other, so that you could end up with a situation like this, where both the brassica and raffinus chromosomes are on one side, and the other side or the other daughter cell receives no chromosomes, leading to aneuploid gametes. And this implies, since aneuploid progeny created by aneuploid gametes are usually not viable, this hybrid of brassica and raffinus is going to be sterile or infertile. However, in the experiments by Karpachenko, there was at one point a spontaneous doubling of the chromosome number, so that the chromosomes were now doubled to 36, so that we have 2 of the brassica chromosomes and 2 of the raffinus chromosomes, and the pair of chromosomes which results from the doubling can synapse with each other. For example, you would have the 2 brassica chromosomes synapsing, and you could also have the 2 raffinus chromosomes synapsing, and these would now segregate reliably, ensuring that the gamete will have N1 plus N2 equals 18. So a new haploid number of chromosomes, and would be able to produce viable progeny. And this is how allopolyploidy works, is that first there is a hybrid between 2 species, and second there is a spontaneous doubling. And it doesn't have to be just double. It can be 2 or more, and that allows synapse and pairing of chromosomes during meiosis in the hybrid, and reliable segregation, allowing this allopolyploid to be fertile and be able to propagate itself. One interesting thing about this allopolyploid between brassica and raffinus was that it could no longer be mated to produce fertile progeny with the either brassica or raffinus. For example, if one takes the hybrid, which has 2n plus n1 plus n2 equals 36 chromosomes, and brassica, cabbage, which has 2n1 equals 18 chromosomes, the gametes will have n1 plus n2 equals 18 chromosomes, whereas brassica gametes would have n1 equals 9 chromosomes, and the progeny would have 2n1 plus n2 equals 27 chromosomes. However, this is a triploid, and as we've discussed previously, triploids are usually infertile, since they produce aneuploid gametes and aneuploid zygotes that are usually not viable. And what this means is that while the hybrid can be crossed to other individuals of the hybrid species, and they can be propagated, they are no longer capable of being crossbred to the original brassica or raffinus strains, which means by the biological definition of species, this hybrid is a new species, since it is no longer compatible with the original species that were hybridized to produce it. Lastly, let's practice with an example problem. Below are given the pairing of chromosomes during meiosis of hybrids between the New World cotton species Gossypium hirsutum and the Old World species G. thurberi and G. herbaceum. and we have to deduce the number of chromosomes in each species. We see here that we are given 3 crosses between pairs of the 3 species. So when we cross Thurberry and Herbaceum, during meiosis in the hybrid, we have 13 large univalent chromosomes and 13 small univalent chromosomes. When we make a hybrid between Hirsutum and Thurberry, during meiosis in this hybrid, we have 13 bivalents, small bivalents, and 13 large univalents. And in the last cross between Hirsutum and Herbaceum, in meiosis in this hybrid, we have 13 large bivalents and 13 small univalents. So let's just try to make sense of this. And first step is to organize the information. One thing that's clear is the number 13, everything is a multiple of 13. Therefore, the haploid number n is 13. The second thing we notice is that in the cross between Thurberry and Herbaceum, we have only univalent chromosomes during meiosis, which means no pairing occurs, and this implies that both Thurberry and Herbaceum are diploid species. So 2n must be 26 for them. And either Thurberry has large chromosomes and Herbaceum has small chromosomes, or Thurberry has small chromosomes and Herbaceum has large chromosomes. Now, if we look at the cross between Hirsutum and Thurberry, or Hirsutum and Herbaceum, what we see is that there are a total of 39 chromosomes. So there are 13 large univalents and 13 small bivalents. And bivalent means 2 chromosomes are paired up. That means there are 26 chromosomes, 26 small chromosomes and 13 large chromosomes. So the total number of chromosomes is 39. And it is the same in the second or the third cross between Hirsutum and Herbaceum, where we again see that there are 39 chromosomes in the hybrid. And what that tells us is that Hirsutum must be tetraploid. Since its gametes, since 13 of the 39 chromosomes are from either Thurberry or Herbaceum, the number of chromosomes in the hybrid from Hirsutum is 26. And that means if the gametes have 26 chromosomes, the Hirsutum must be tetraploid, having 52 chromosomes in all. Now, the only thing remaining to decide is what is the combination of large and small chromosomes in the 3 species. So let's say that in this cross between Hirsutum and Thurberry, the large univalents, the large chromosomes, are from Thurberry. This would imply that the 13 small bivalents are from Hirsutum, which means that Hirsutum's gametes have 26 small chromosomes, which means Hirsutum has 52 small chromosomes. But we notice then that in the third cross between hirsutum and herbaceum, if the large chromosomes are from Thurbury, the small univalents must be from herbaceum, so that the 13 large bivalents are derived from hirsutum. This contradicts our previous conclusion that hirsutum has 52 small chromosomes, which implies that this scenario where Thurbury had the large chromosomes and herbaceum had the small chromosomes is incorrect. And in fact, it must be that the small chromosomes are from Thurbury and the large chromosomes are from herbaceum. So let's try that out. Now, since Thurbury has small chromosomes, 13 of these bivalent chromosomes are from Thurbury, which means the other 13 are from hirsutum. And these 13 large univalents are also from hirsutum. This implies that hirsutum has 2n1 equals 26 large chromosomes and 2n2 equals 26 small chromosomes. And let's check if this works out in terms of the third cross between hirsutum and herbaceum. Since herbaceum has large chromosomes, it means that 13 of these large bivalents are from herbaceum, and the other 13 of these bivalents, the other of the pair, are from hirsutum, and these 13 small univalents are also from hirsutum, and it checks out. Therefore, interpreting all this information from the crosses leads us to conclude that Thurbury and herbaceum are diploid species, where Thurbury has a haploid number of 13 and it has small chromosomes, whereas herbaceum also has a haploid number of 13 but it has large chromosomes, while hirsutum has 26 large and 26 small chromosomes, making it a tetraploid. And what must have happened is that hirsutum must be an allopolyploid formed by the hybridization of Thurbury and herbaceum, followed by chromosome doubling, allowing hirsutum to be to produce viable progeny and become a new species.